Welcome back to BOGObiology. This screencast is going to be about the concepts of genetics and heredity. For the purpose of this screencast, we will be using paint cans as a metaphor for an organism. Pay careful attention to the label on the can as well as the color of the paint because this will help you to understand some of the key terms. To begin, we're going to define two terms that many people find confusing: genotype and phenotype. The genotype is the set of genes that an organism carries in its DNA. These two paint dots depict what components are found within each can. The phenotype of an organism is its observable characteristics. It's what we would see when we opened the can of paint. Once you open the can of paint, you can see its phenotype, i.e. what color the paint is. The first two cans are pretty straightforward. The can with the blue genotype has a blue phenotype, and the can with the yellow genotype has a yellow phenotype. But what about the can on the right, whose genotype contains both blue and yellow components? This can is called a hybrid. Its phenotype is blue because the blue paint overpowers the yellow paint. Even though both paint types are present in the can, i.e. the organism's DNA, only the blue paint shows up in the phenotype. Only one unit of blue paint is needed in order to determine the color of the paint in the can. This makes blue the dominant color. Dominant genes overpower others in order to determine the organism's phenotype. Recessive genes require two copies to determine the organism's phenotype. For the paint to be yellow, the can needs two yellow components because yellow is the recessive color. In short, if any blue is present in the can, it will overpower the yellow. There are multiple variations of each gene, and we call these varieties alleles. These variations arise via mutation or mistakes made during the reproduction process. In order to easily describe an organism's genotype, we use two terms, homozygous and heterozygous. When an organism is homozygous, it has two identical alleles for a given trait. For our purposes, the trait in question is color. Notice that an organism can be either homozygous dominant or homozygous recessive. When an organism is heterozygous, it has two different alleles for a given trait. A heterozygous organism can also be called a hybrid. A major reason to study genetics is to be able to predict the probability that two organisms will produce offspring that have certain characteristics. Provided that you know the genotype of both parents, it's possible to predict the probability that their offspring will display certain traits. For our first example, we'll be breeding a homozygous blue paint can with a homozygous yellow paint can. We'll put one parent's genotype here, and the other parent's genotype over here. Now your Punnett square should look something like this. If we assume that these are monogamous paint buckets, the only possible genes their offspring can have are those from their parents. Each parent will contribute one allele to their offspring. We can use this diagram to figure out the possible combinations of alleles that their offspring could have. In this scenario, because one parent has only blue alleles and the other parent has only yellow alleles, all of their offspring will have one blue and one yellow copy of the paint color gene. When we fill them in, these are the possible genotypes for the offspring. We have 0 homozygous dominant offspring, 4 heterozygous offspring, and 0 homozygous recessive. Now let's take a look at the phenotypes. I find color coding my Punnett squares to be helpful, so I'm going to shade in the phenotypes for both parents first. After shading it, the Punnett square will look like this. When we've shaded in the phenotypes for the offspring, we can see that all of the offspring will have the same phenotype as well. All 4 offspring will have the dominant phenotype and none will have the recessive phenotype. All of the offspring from the first pairing had the blue phenotype, but each one of them was carrying a copy of the recessive yellow allele. In addition to calling heterozygous organisms hybrids, we also sometimes call them carriers. These organisms may not even be aware that they are carriers for the yellow coloring because their phenotype is blue. However, the yellow possibility is still there in their DNA and may come out when they reproduce. Before I fill it in, see if you can figure out the possible genotypes and phenotypes for the offspring of 2 heterozygous paint cans. These parents could have offspring with genotypes that were homozygous dominant, heterozygous, or homozygous recessive. They have a 25% chance for offspring with a homozygous dominant genotype, a 50% chance of heterozygous offspring, a 25% chance of homozygous recessive offspring. Instead of having all offspring with the blue phenotype, there is now a 25% chance that they could have offspring with the yellow phenotype. It's worth noting that the probability starts over every time they reproduce. Having one blue offspring doesn't increase the likelihood of getting a yellow offspring the next time around. To finish up, we're going to tackle a very common problem, which is how to determine the genotype of an organism if you don't already know what it is. The first step is to determine the phenotype of the unknown organism. Obviously, it's going to be useful to do a little research to figure out what the different phenotypes look like. If the organism has the recessive phenotype, you can stop there because you know that it's going to have the homozygous recessive genotype. If it has the dominant phenotype, you have two options. The genotype may be homozygous dominant or it may be heterozygous. If the organism has an intermediate or a blend of phenotypes, unfortunately that means that you probably have a case of codominance or incomplete dominance, and traditional Mendelian genetics rules don't apply. We're going to proceed as if one of the first two scenarios is the case. Next, you're going to perform a test cross. You're going to breed the unknown organism with a homozygous recessive organism. Then you're going to analyze the percentages of offspring phenotypes. Based on the offspring phenotypes, you can work backwards to take an educated guess at what the genotype of the parent is. The more offspring you can produce, the more accurate your percentages are going to be. The smaller the pool of offspring, the more likely your data is to be skewed. This technique is also much more efficient if you're using it to analyze organisms that reach maturity and reproduce very rapidly. In example 1, the unknown organism has the blue phenotype. To determine its genotype, first ask yourself the question, what are the genotypes of all the offspring? We know that every one of the offspring is going to have at least one yellow allele from the yellow parent. However, because all of the offspring also display the blue phenotype, they must have a blue allele as well. This means that all of the offspring are heterozygous. Now try and work backwards to answer the second question and work out what the genotype of the blue organism is. If you worked out that the unknown organism was homozygous for the blue allele, you're correct. Now see if you can solve this problem on your own. If you're following along, this is what you should have written down. Because these two offspring have the yellow phenotype, they must have inherited a yellow allele from each of their two parents. For this unknown organism to have contributed a yellow allele but still be blue itself, it must be heterozygous. A few final things to keep in mind. This screencast is a simplified overview. Mendelian genetics are complex. Mendelian genetics are also not a foolproof system. There are very few one gene to one trait matchups. Most traits are determined by the interaction of several genes, and there are also cases where one gene determines several different traits. Some traits are also tied to the sex of the organism, and there are also environmental factors to take into account. Additionally, in many hybrid organisms, they display either a blend of dominant and recessive traits, or they show aspects of both traits simultaneously. If you're interested in a future video about epistasis, pleiotropy, codominance, incomplete dominance, and related topics, please let me know by leaving a comment in the field below. Thanks for watching, and don't forget to subscribe.